just make up a sequence here. Right? So there's our top strand. We'll make a bottom strand. C, A, C, T, G, A, G, C. Generic DNA sequence. You go to make RNA. Is RNA going to use both strands or just one?
Just one.
Just one. And is RNA made 5' to 3' like DNA, or is it made the opposite way? Remember which N new nucleotides are added to with RNA? It's the same as DNA, what would that be?
5'.
Not 5'. Go with the other one. So we're going to add new nucleotides to the 3' end of the RNA. So, two possibilities here. Right? These two strands split. Right? So I'm just going to separate them. Your two strands separate. If you get RNA to this one, it's going to start here and run that way. Right? So we're going to add to the 3' end. So you'll have C, G, A, G, and instead of T, what do you get? U, right? This is RNA, so we got to put in a uracil. C, A, C. That was if you use the top strand. If you use the bottom strand, you're going to get a completely different sequence. Going to start 5', going to run that way. G, U, G, A, C, U, C, G. Right? So that's the first thing to remember. Only one of the two strands is going to get copied. The strand that is being copied is called the template strand. Okay? So this would be your template strand. And this one would be a template strand. Does anyone know what the other strand would be? Right, because DNA is double-stranded, so there's really a second strand here. What's that other strand that's not used as the complementary strand? Has anyone learned that term? It's called your coding strand. It's called the coding strand is because it has the exact same sequence as the RNA strand. The only difference is you have to replace the T's with the U's. Okay. A little bit more about transcription, right? Copying DNA to RNA is transcription. What enzyme or enzymes does this? Remember the name of the enzyme or enzymes?
RNA polymerase?
It's RNA polymerase, all on its own. There's no other enzymes you need to know. So, I'm going to draw a generic one here. Oh, come here. You have your DNA. And you have your actual gene right here. Right, this is DNA. And RNA polymerase is going to bind the DNA in front of the gene. binds DNA in front of that gene. Does anyone remember the name of the place RNA polymerase binds? It's a very specific location on the DNA. Right here. So we're going to call that the promoter. So RNA polymerase binds the promoter. It actually separates the two DNA strands. So it does what helicase would normally do. And then it does what DNA polymerase would normally do, and it'll run along here and make the RNA transcript. And then there's a stop point. Put a little more space down here. This is going to be its stop point. I don't remember what the stop point is called. Bunch of movies by that name. The 80s movies, an old Schwarzenegger movie. The Terminator. That is the Terminator. They got a million of them now. I don't know who's all in them. But that's your Terminator sequence. Huh?
What did you say that promoter did?
That's where the RNA polymerase will bind to start transcription. And it's going to bind upstream of the gene. So if you haven't learned some other terminology, this direction is known as upstream, and this direction is known as downstream. Oh. If there was just a promoter, all of the RNA would be transcribed all of the time. So the cell is going to have other proteins that help RNA polymerase bind, called transcription factors. Right. So transcription factors help the RNA polymerase bind. And we will talk more about those at the end of the day. Most transcription factors in bacteria are what are called sigma factors. That's just their fancy name. Sigma, if you know the Greek, a sigma factor. In eukaryotes, they have very different names. All right. Let's look at the notes and see what else we might have missed. Come here. Okay. So, here's a nice pretty drawing, much better than my own. So RNA polymerase is going to do all the work. It's going to bind at the promoter on the DNA. It's going to make a copy of one of the two strands of DNA, and that all depends on where that promoter is. And it'll keep making that copy till it hits that Terminator sequence. It's always going to add to the 3' end, and unlike DNA polymerase, this does not need a primer, so this can just run. Questions on that? Okay. I'm gonna hit your memory some more. In eukaryotes, we have this stuff called post-transcriptional modifications. So you remember, you get this primary RNA transcript in eukaryotes. Where's that at in the eukaryote? Where in the cell? Where's the DNA in the cell?
The nucleus.
The nucleus. So that's also where the RNA is. Prokaryotes don't have a nucleus, so where's the RNA in a prokaryote?
Cytoplasm.
The cytoplasm. Okay. So eukaryotes, that primary transcript is in the nucleus. It's going to get modified. Does anyone remember any of those modifications? there's something that happens to the 5' end. Go ahead, Sarah.
Isn't there like a methyl cap and a polyene too?
That's right. There's a methyl cap. I usually say a methyl guanine cap. At the 3' end, you get a poly-A tail. So what this means, 5' end, you just get a modified guanine added. 3' end, you get a whole bunch of adenines thrown on the end, usually 100 or 200 adenines. And then there was one other thing that happens in the eukaryotes. Go ahead.
Is like the introns and exons or whatever?
Yeah, splicing. So introns are removed and the exons are hooked together. What you will love about the prokaryotes, they don't do any of this. Right? What comes off of this transcript, that's it. That is the final transcript. So this is a eukaryote only process. We will use that later. So we need to know it now. And then in the eukaryotes, that methyl cap will take it out of the nucleus. All right, let's see if we're missing anything else. Yeah. So if you miss any of the terms, template strand, coding strand, place your uracil with thymine. I'm just making sure we didn't miss anything here. The terms upstream, this way. Downstream's gonna be that way. Okay. Before I go ahead, because I know there's one more thing in there. There is one thing prokaryotes do different than eukaryotes. So a eukaryote, they're going to have a promoter and one gene and then a terminator. What you're going to see with the prokaryotes frequently here is we're going to have our long DNA molecule. And we're going to have our promoter. And we're going to have multiple genes in a row. So you're going to have gene A, and they're always lettered for some reason. Gene B, gene C, and so on and so forth. And then you'll have your terminator. This is what's known as an operon. When you have a bunch of genes under the same promoter, this entire structure is an operon. And we'll look at those quite a bit, probably at the end of today. And so what happens is your RNA polymerase sits down, and it makes one really long mRNA. This is what we call a polycistronic RNA. So that's something our eukaryotes do not do. It's something that's specific to the prokaryotes, and actually a lot of viruses do that. Questions on that one? What we're mostly looking at is similarities and differences. So this is more about transcription, just the process. Elongation, termination at the terminator. And then we get to our polycistronic RNA. Now when it goes to do translation, it will translate each gene separately on this single mRNA. The benefit of that, though, is let's say all of these genes encode proteins that are part of the same metabolic process. So I make protein 1, I make protein 2, I make protein 3. And the cell gets the same amount of each of those proteins. So they're all coupled together. So these tend to be genes that make proteins that are involved in the same metabolic process. And we'll see that again in a few minutes. The other thing, do I have that back there? Okay. No, that's just terminology. So, I really thought I had this in here. If you haven't learned this before, terminators are usually kind of cool. They're usually a bunch of ATs, and what they do is they come up, and the RNA will actually bind to itself, and it makes what's called a hairpin loop. And that'll actually what stop this. But I must not have it on the exam because I didn't put it in here. Okay. Let's move on. So there's just a short list of the components. Negative strand DNA, which is the template. Positive strand DNA, here's the coding. And then our promoter, our RNA polymerase, and our terminator. Hopefully that's pretty simple. Let's get to the hard part. Let's see what we can remember about translation. What you learned about translation in the past is actually what E. coli does. We don't know of a lot of differences in eukaryotes. So the translation we teach is E. coli's translation. So that makes that easier. But let's just start with an RNA sequence. Okay? So we've already done transcription. This is going to be RNA. And we're going to do translation. And we're going to start with AUG. If you don't know why, we'll talk about it in a minute. We need to make up some 3-letter codes here. So let's do GGG, CUA. I have no idea. I'm just making these up. GAA, CUU. There we go. Okay, so there's an RNA sequence. And you can always just make these up. Why would I be making these up in 3-letter sequences? Remember what those 3 letters are called?
A codon.
Right, these are going to be the codons. So that is what the cell is going to read to convert it into an amino acid, are those 3-letter sequences. This codon to amino acid change, what we call the genetic code, is pretty much universal. So what E. coli uses is pretty much what you use. That's why we can make insulin cheaply, because we took the human gene for insulin and put it in E. coli. And E. coli now pumps out insulin. They say we can make it cheaply. All right, just a little bit. I am going to have you do one of these on the exam. Do an actual translation. It will give you the codon table. So you just have to be able to use it. All right. There are 64 possible codons. 3 of them are stop. Stop just means there's no amino acid that encodes for that codon. And so that's where protein synthesis stops. There is exactly 1 start codon normally, and that's AUG, which is why I started there. Now, some bacteria are jerks and will start at GUG. We're not going to worry about that right now because your book doesn't really show that. But a few bacteria will use GUG to put in a methionine because they're idiots. But we're going to use this table. I'll give you a nice simplified version of this. You don't need to know the long names of the amino acids or any of that stuff, right? We'll have those little 3-letter codes. So we're going to always translate 5' to 3'. Right? So we're going to always translate this way, 5' to 3'. AUG is going to insert what amino acid?
Methionine.
Methionine, or we're just going to put met. This is not going to have a 5' end or a 3' end because it's not a nucleotide. I'm going to go back again. Does anyone remember the structure of an amino acid? Your central carbon, 4 groups around it. Who wants to help me out? Now you got it.
I'm like, I know this.
I know you know this. You've probably been tested on this 20 times. Anyone else? There are 4 different groups around an amino or around a central carbon here. Go ahead, Sarah. There's an amine group.
Is there a hydrogen?
There's a hydrogen. Awesome. We got 2 of 4. We need 2 more.
Oxygen.
Not as simple oxygen.
Isn't there an R?
Yep. So this is what makes them all different.
Is another carbon?
So there is another carbon. It's part of a group. So this is the amino part. What's the acid part?
Probably like something
Carboxyl.
that, yeah.
Yeah, carboxyl. Why am I telling you this? Proteins have ends, just like DNA and RNA have ends. All right? The front end here, as I'm going to call it, is the amino end. So this is where you're going to find your NH3. We often just call it an N-terminus. So nucleotides have the 5' and 3'. Proteins don't have 5' and 3'. They have amino and carboxyl. All right. Let's do the rest of this translation. GGG. I have no idea what is GGG. What is it? That glycine over there. Glycine's the fun one. He's got another hydrogen down here. It's the one I know pretty well. CUA. What is CUA?
Leucine.
Leucine. So I will have you do one of these on your exam. What's GAA?
Glutamate.
What is that? GLU? Yeah. And we got one more. CUU. CUU would be
Leucine.
another leucine. And if this N is an amino terminus, what's this N? Your amino acid.
The decarboxyl.
Carboxyl. This is our carboxyl terminus. C double bond O OH, or we'll just call him a C terminus. Okay. So proteins have direction too. All right. Now comes the harder part, is how does the cell do this? Okay. Yeah. So nucleotide sequence is going to determine the amino acid sequence. That's easy. First thing that's going to come in is your ribosome. What was the difference between prokaryote and eukaryote ribosomes? Does anyone remember? You get the small subunit of the ribosome. What does it say is the prokaryotic ribosome? Your prokaryote is a 30S small subunit. Remember what the eukaryote was? So this one was a 40. The small subunit was 40, yes. When we start talking about antibiotics, we're going to target these ribosomes a lot. Right. So the small subunit of the ribosome is going to come in. It's going to bind what's called a ribosome binding site. Or an RBS. And that is going to be upstream of that AUG. It's usually not very far upstream. It's usually like 10 base pairs upstream. Oh, there we go. So it's going to bind. There we go. The next thing that comes in is a tRNA. So after your small subunit comes in, a tRNA binds the RNA. Remember your tRNAs, they're very small RNA molecules that have folded back on themselves. I'm just going to draw it. That's a horrible idea. All right. It's folded back on itself. It's going to have an anticodon right here. And the anticodon is complementary to the codon. So across from A, it would have a U, an A, and a C. And we still have to do that same 5', 3' anti-parallel. So if this ends 5', this ends 3', and ends 5'. And attached to that anticodon will be the matching amino acid. So this will have a met attached to it. Right? That's where our tRNA binds our RNA. What type of chemical bond would that be between complementary nucleotides? A hydrogen bond, right? A pairs with T with a hydrogen bond. G pairs with C with hydrogen bonds. So we have a small subunit. We have the mRNA. And we have a tRNA. The third thing that's going to happen is this large subunit comes in. And that tRNA is going to go in the middle site. So, a large subunit of the ribosome places the tRNA in the P site, which is the middle site. And if we're going to do the sizes again, go back. Our prokaryote has this way, 50S subunit. What does your eukaryote have? Anyone remember? Just go up 10. So this will have a 60S. Right. So the ribosomes look a little different. That's how we get it started. If you ever wonder what the tRNA kind of looks like more accurately, this is more accurately what a tRNA looks like. It's a better image of that. All right, so here I'm just going to walk through it real quick, and I'm going to show you a video of this because I think it's easier to see it in motion. So here's the beginning. first tRNA with the methionine here. Bacteria use a formal methionine for their very first methionine because they're different. It's in the P site. It's in that middle site. P means peptidyl. It's where the peptide bond is created. So that's the enzyme part of the ribosome. Your second tRNA is going to come into the A site. It's going to have an anticodon, complementary to the codon. Once that second tRNA is in there, the ribosome uses a little bit of ATP and makes a peptide bond between those two amino acids. Oops. And in the process of building that bond, it breaks the bond between the first tRNA and its amino acid. Right? So your growing peptide chain now is in that A site. And when that happens, ribosome shifts. And it shifts exactly 3 nucleotides. The third site is the E site. It means exit. Those tRNAs don't have any amino acid attached, so they exit and they get recycled. They actually will get another amino acid attached to them, and they will be reused over and over. A site's now empty, so a new tRNA can come in. The whole thing starts over. It's slow, it's expensive, costs a lot of ATP. So the cells don't do this when they don't have to. That will go on until it hits a stop codon. So if you like a quick summary, your book has a nice summary there. But it'll go on until it hits the stop codon, and when it does, everything dissociates. Now again, bacteria have this polycistronic RNA. The ribosome will read one. It generally doesn't even dissociate. It actually will release that peptide chain because of the stop codon, and then it'll start on the next one. And then it'll release that polypeptide chain because of the stop codon, and then it'll read the next one. So that's the benefit of this polycistronic RNA. Okay. Questions on that? Hopefully a lot of that's review. Hoping somewhere that information is still there. Before I show you the video, the one really, really amazing thing about prokaryotes, because everything is in the cytoplasm, and because there's none of these modifications. So here's your DNA. The cell is making RNA. The moment there is a ribosome binding site available, a ribosome will bind it. And it's going to start making protein before this RNA is ever even finished. So bacteria can usually ramp up protein production in a matter of minutes because of the way they can couple transcription and translation. And you definitely can't do that in a eukaryote. You can see these under the microscope, a good electron microscope. We call, when you see one of these structures with lots of ribosomes, we call it a polyribosome because there's a lot of them together. And I don't know if your book someone has a really great electron micrograph of this. I don't know if I put it in here. I didn't. Okay. Promised you a video because that's a lot to think about. Let's see it in motion. You can always watch these. This one is for eukaryotes, so it will have the modifications being done. I don't have a micro one. Oh no, they're doing their chanting.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
So hopefully seeing that in action helped a little bit. Another kind of interesting tidbit, so at the part where the protein was produced in the ER, you know, the protein started being produced and then it docked to the ER and the rest of it was made, that's actually in a bacterial cell. If the protein has that start, again, what we call a signal sequence, it actually will dock with the cell wall, and that protein gets secreted from the cell. So the way that our cells direct things to the ER, bacteria will actually use that to direct those proteins for secretion. So, another little tidbit. Okay. So hopefully most of that was review again. Questions on translation. Now we're going to get to where it's really different. The way we regulate expression. Not going to discuss eukaryotic gene regulation because that's a whole class, like a whole semester class in and of itself. So we're going to talk about prokaryotic gene regulation, which is very simple, actually. We're going to go back and make an operon image again. And take this down. Let's draw another very simple operon. Oh my goodness. All right. Here's our DNA. 5 prime, 3 prime, 3 prime, 5 prime. And we're going to have our promoter, just like we have. And we'll have a few genes. All right. And we'll have our terminator here. So we'll have A, B, and C for now. Okay. So we already talked about the post-transcriptional modifications here and moving things to cytoplasm and the different ribosomes. So if you missed any of that, it's kind of summarized for you. gonna look at these operons and how they are regulated. And there's really three possibilities. Right? We can have something that is constitutive, which means it's always transcribed. So generally, the enzymes used in glycolysis are expressed constitutively. They're always being made. Right? We have operons that are repressible. I can write R-E-P, repressible. These are going to be usually on, but they can be turned off. So these are usually transcribed. But they can be turned off. And we're going to see these in the case of anabolism, when the cell is creating something. What it's going to do is it's going to create it till there's too much of it built up in the cell. And then it'll turn this off. All right, so we usually see this for anabolism. And what we will actually look at as an example is the operon used to make the amino acid tryptophan. I call this the trip operon. The trip operon is used, they're the proteins that are used to make the amino acid tryptophan. The alternative is inducible. These are usually off. But they can be turned on to transcribe them. And whereas our repressible were anabolism, these are usually catabolism. So glucose is always broken down. But what we're going to look at is what happens when the cell gets fed lactose, like on McConkey's media. Then it's going to want to turn on the proteins, or make the proteins, to break down that lactose. Usually catabolism, and our example here will be the lac operon. Thank goodness the Amoeba sisters have actually made a video on this operon. So if you want there's an extra credit to watch that one with the Ned puzzle. So this is kind of confusing. All right, I have 3 more minutes. I'm going to use them to get started. So here's that summarized for you. All right. Okay. So we're going to look at some specific DNA binding proteins called repressors. Repressors bind the DNA and stop transcription. Where's my green one? Okay. Well, if we're going to stop the RNA polymerase, we want to sit down between him and the genes. This is where our repressor is going to bind. It binds to a site called an operator. I guess I do have that written in there. I just didn't underline it or bold it or anything. Okay. So we are going to have right here a DNA binding site called an operator. It has to be between our promoter and our genes. Okay. Going to do a quick introduction to this and then we'll review it on Monday. In fact, instead of me showing you these, instead of doing this as text, because I love these little super simple videos that they used to have with textbooks. I don't know if they're still in your book or not. So if we go down here, so we have these 3 operons. Just a general overview. Let's watch the little induction one. Now these are incredibly simplified. Here's a little 2-minute how induction works.
The life operon of E. coli includes a promoter, an operator, and 3 structural genes encoding proteins involved in the catabolism of lactose. The lac operon is an inducible operon, meaning that the genes are usually not transcribed. When lactose is available to the cell, the operon is induced or stimulated to produce the proteins of lactose catabolism. The E. coli chromosome contains a regulatory gene near the lac operon. This gene is constantly transcribed and translated to produce repressor protein.
Okay, so let's add that to our drawing really quick. So that's upstream. So you're gonna have another promoter here. And you're gonna have another gene. And these are usually labeled I. I have no idea why. Right? This promoter is constitutive, meaning it's always running. And so it's always making RNA, which is then always making the repressor protein. Oops. So any operon that is repressible or inducible will have this associated regulatory protein. Okay, we'll watch the rest of this. Be a minute and a half.
The
Oh,
repressor
good.
protein binds to the operator DNA of the lac operator, physically preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for lactose catabolism cannot be transcribed into mRNA. When lactose is present in the cell, some lactose molecules are converted to allolactose. Allolactose binds to and inactivates the repressor proteins, preventing them from binding to the operator. The structural genes are transcribed, resulting in the production of the enzymes needed to catabolize lactose. Since allolactose induces or stimulates transcription of the lac operon, it is called an inducer. Notice that the inducer acts by turning off the repressor, not by actively stimulating the genes to be transcribed. When lactose is not available, the repressor proteins are active. The structural genes of the lac operon are not transcribed. This control prevents the cell from producing the proteins for lactose catabolism when lactose is not available.
And that is that. We'll go over this again on Monday. And we'll add a little more to it. But there are 2 videos there, one for induction and one for repression, if you have a couple minutes to watch.